Hi, everybody. Thanks very much. So, as you said, my name is Charles Stacey. I'm the president and CEO of Cerecin. Thanks very much, John. And I'm going to tell you a little bit about our science, but also a little bit about our business model, which is, I think, intentionally disruptive. I mean, and differentiate in terms of thinking. I think a lot of what we've talked about over the course of the last 1.5 days has been that we're pioneering this space, and there's a lot of change happening. And I think that new models and structures that we have incorporated within our development as a company are reflective of that. So I'm going to tell you just about our lead program in Alzheimer's disease. Alzheimer's disease, of course, needs no introduction. Suffice to say that it is one of the biggest unmet clinical needs of our time. The biggest risk factor is, of course, aging. Last drug compound approved in 2003, which combined with a high number of failures in the space really makes for the perfect storm within Alzheimer's disease. The map on the right-hand side explains part of our strategy, which I'll elaborate on later, which is that the majority of the world's aging population and the majority of Alzheimer's patients, accordingly, are focused in Asia, and it's now where I'm based, and the company is headquartered. So some of you would have seen this graph on the right-hand side. What it shows is the biomarkers that develop within the brain as patients develop Alzheimer's disease. The vertical dotted line at time point 0 shows the point at which Alzheimer's patients are diagnosed, around 65, 70 years old. And what it shows is that 10, 20, 30 years beforehand, if you're at risk of developing Alzheimer's disease, these changes are already happening within the brain. You see the increase in in amyloid, in tau, and the decrease in glucose metabolism that occurs within the brain. And the images on the right-hand side, this is some work published by Eric Ryman, show that this is FDG PET scans of patients with Alzheimer's disease. The purple areas of the brain there show the decreases in glucose metabolism that occur within the brain when a patient has Alzheimer's disease. The blue areas are the parts of note, and what that shows is that in a patient, and Eric Ryman showed that at a mean age of 30 years old, you can identify this decreased glucose metabolism already starting within these patients, and that gradually develops throughout the course of the next 3 decades until that patient manifests as having the signs and symptoms of Alzheimer's disease and is diagnosed. So in line with a lot of what we've been discussing over the last day and a half, there is this opportunity to intervene early with the right sort of therapeutic modality and intervene during this period of 30 years or so that exists within Alzheimer's disease in the prodromal phase. So a little bit about our compound and the science underlying. So what's well recognized is the brain is highly metabolic, and the slide previously showed that there's this decrease in glucose metabolism that happens, is progressive and regionally focused within the brain. But the brain, although it's only about 2% of the body weight, accounts for about 25% of the body glucose utilization. So it's hugely metabolic, and therefore you imagine that if there's a problem with glucose metabolism within the brain, it's a massive problem, and ultimately these cells become stressed, and ultimately the cells die. But fortunately, the brain has a backup mechanism. The brain can utilize 2 different energy substrates, both glucose as well as ketone bodies. And ultimately, what we ask as a company is, if the brain in Alzheimer's disease has a problem metabolizing glucose, can we provide it with ketone bodies and therefore restore metabolism and prevent the decline that occurs in Alzheimer's disease? Essentially addressing the upstream effect here, and what this shows is essentially that our compound, caprylate triglyceride, is addressing the upstream problem of impaired brain metabolism, which is upstream of amyloid and of tau, which potentially explains why a lot of the drugs that have been developed addressing these targets haven't worked as we'd expect them to. And this analogy has been used by some of the others in the audience, but essentially think of the brain as a hybrid engine, can utilize both electricity as well as petrol, and therefore if one of those components is lacking or is dysfunctional in some way, can you provide the other energy substrate and therefore restore function? And the way we do this is by providing a very specific structured lipid, a lipid called a medium-chain triglyceride, caprylate triglyceride is the name of our compound, and we provide a very specific proprietary formulation of this compound to patients, which is then taken into the systemic circulation and then broken down by the liver. The fatty acids are broken down by the liver to provide ketone bodies, flooding the circulation and therefore providing this alternative energy substrate. And over the course of the last decade, we've set out providing the clinical evidence to support the efficacy of this compound. This shows some of our data from our phase 2b study. The graph on the left-hand side shows the treatment arm in blue, on the right-hand side shows the treatment arm in blue and the placebo arm in below. And what you can see here, within 45 days, we're showing a difference of about 3.5 points on the ADAS-Cog score, the gold standard endpoint for cognition, which persists out to 90 days. And when you do a washout period and you remove the therapeutic agent, then that decreases over time. This also shows some imaging data that was done by UCLA and was published last year. This was an elegant study which showed, which labeled oxygen, so it used an O15 labeled labeled water. And what this attempted to show was increases in cerebral blood flow as a surrogate of cerebral metabolism. So they took two groups of patients, placebo and control, placebo and treatment group. The treatment group they treated with our caprylate triglyceride for a period of 45 days. And on the right-hand side there, you can see the images. The red parts of those brains show areas of increased cerebral blood flow. What you can see there is that it particularly affects areas of the brain associated with memory, so temporal and parietal lobe. You can also see that there's an effect with, particularly affects the ApoE4 non-carriers. This is something we've seen within our last couple of studies, but also has been shown by third-party groups. And you can see both an effect after a single dose, but also that effect is larger at 45 days. So just to highlight as well that some of the nice third-party validation we've seen has been when groups have done studies in the ketogenic diet. Robert Kokorian showed this first in 2012. Studies have since been done by Russell Swerdler and Suzanne Kraft, and essentially what they've done is they've said, look, if there is an opportunity to induce ketosis by another mechanism, of course the answer is yes, you can use a ketogenic diet. And for those of you that know, that don't know, this is an incredibly low, difficult diet to follow, low carbohydrates, low proteins, and high levels of fat. But if you can get patients to comply with this diet, as they did in these studies, you can show that there is an improvement in cognition within these patient groups. Unfortunately, all of these studies, whilst they're able to show an improvement in cognition, they also conclude that this diet is incredibly difficult to follow and comply with, especially when you've got an autonomous elderly population. And therefore, but this is nice support of our mechanism, because ultimately what we're doing is drugifying the ketogenic diet and allowing patients to maintain a normal diet, yet still enter a state of ketosis. So the last comment that I'll make is this one around business model. So Alzheimer's disease, as you all know, has had a huge an unparalleled failure rate associated with it. The numbers quoted by Jeff Cummings were of 99.6%. So we're clearly doing something wrong as an indication. And therefore, at Cerecin, we believe that given the challenges and given the different approach that we need to look at things differently. So the target we address is differentiated. As you know, most of the drugs that have been developed in this space have addressed amyloid and increasingly addressing tau. And of course, that's not what we address. We're addressing this metabolic deficit, which we believe is upstream and fundamental to the disease process. So in terms of platform, we take a holistic view to drug development. Our focus currently is on our drug development of tricaprolin, which is now entering phase 3 and is addressing mild to moderate Alzheimer's disease. But our compound that we use, being a structured lipid, is GRAS, and there is definitely the potential with time, once it's established as an approved drug, to then move into the preventive stage and to move into the 10th, 20th, the 30 years before the disease manifests, and potentially go down non-drug avenues and to look at some of the sort of other regulatory categories that can exist, and will be open to us having a GRAS compound. We're also differentiated in terms of the partners we've chosen. So our biggest investors and partners to date are both the Nestle Group, so the world's largest nutrition and food group, but also last year we brought on board alongside them Wilmar, who are Asia's biggest food and agri conglomerate. And I think if we recognize the shifts that we're seeing in healthcare, if we recognize the changes that are afoot, then these sorts of groups are the ones that we think are going to be increasingly important when addressing earlier stages of disease and addressing some of these preventable, looking at prevention rather than, let's say, treating and managing disease. And they bring huge expertise and very useful insights to us as a company in challenging some of the norms that we've established and accept within pharmaceutical and drug development. Development strategy. So whilst we recognize that the US is a hugely important market, we are also looking at other markets. We recognize that global drug development now is global by nature, but also there are other regulatory frameworks and other regulatory agencies that do think about things differently. So we look at these, and we just moved our headquarters last year to Asia for exactly this reason. And we recognize that we're engaging with these other agencies and developing drugs that don't just treat the 4 million Alzheimer's patients within the US, but rather the 40 million patients globally is a critical piece of this. And finally, with regards to location, we were based in the US for the last 10 years. We set up last year in Singapore, and we're building a team there for exactly this reason. Thanks very much.